This is the third in a series of videos where I describe how to perform ChIP-seq analysis using Galaxy. In the first video I talked about how to use Galaxy and gave a brief introduction to what ChIP-seq analysis was. In the second video I used CEAS to perform peak calling on the data that I'd prepared for this tutorial. In this video I'm going to be talking about how to get some extra information which is features the genome that binding regions are close to. And to do this I'm going to use a tool called CEAS, which stands for the CIS Element Annotation System. To find this in Galaxy we can use this search tool and type CEAS and click on that. Okay, so it requires from us a bed file of ChIP regions. So we have all this from Max in our history already. I'm actually going to provide it with the summits file. Now when you provide binding regions to CEAS, what it actually does is it finds the midpoint of each one and then looks to see how close that is to a set of annotated genes. So by providing it the summit file, you're providing it with the location of the peak of binding, the highest pile up of fragments in the binding regions. I think that's slightly more accurate use. Okay, so that is number 4 in our history. Okay, now also it will take a WIG file for looking to see where binding is greatest in terms of fragments over different areas of the genome, in particular in relation to genes such as upstream, downstream, intronic, exonic, etc. Now we have these WIG files from our Max analysis again, so we can provide it with the treatment WIG, which is our ChIP data. Now we're not going to in this tutorial, but say you had another bed file of interesting regions, for example lists of CPG islands or other areas of interest that your laboratory is interested in. You could upload that data into Galaxy and input it here, and it would also look to see where binding fragments in the form of this WIG file occur over your regions of interest as well, in addition to the binding regions. Okay, we have to tell it what genome we're interested in. In this case that is mouse MM9. Now these other parameters you can quite happily use the defaults. That will give you a reasonable result, but what I tend to do is change these values for what it considers to be the promoter region, so a lower, middle and upper region, which equates to sort of basal binding right near the TSS to very distal events away from it. So this is just me, but I tend to use 500, 1000 and 10,000 as the limits of the different portions of the promoter. Okay, so that's all the parameters we need for that, and then we can press execute to start the process running. So again we have the little clock showing that it's waiting for entry into the queue. Now the blue spinning circles show that it's entered the queue and is working. So let's just briefly describe what CEAS is doing. So with CEAS we're able to see where our binding regions and our non-redundant set of fragments from MAX are distributed relative to features of the genome. Principally we're interested to see where they sit in relation to genes in the form of ChIP-seq annotation, and any other feature that's uploaded by the user in the form of a bed file. So ChIP-seq is a set of non-redundant and well annotated reference genes. So it's a relatively small set, but can be relied upon. Next let's go back and see how our CEAS analysis is getting on in Galaxy. Okay, so CEAS has now finished running as the history items have gone green. You can see there's two output files. There's a log file here. It doesn't really give an awful lot of interesting output, apart from the fact it tells you what parameters were used, which is quite useful in the future. The interesting thing for us is this PDF report. You can see this by clicking on the eye. Now this first section would normally give you an indication of whether there's any difference in distribution between the binding regions you've supplied and the number that's expected to be present across the whole genome. However, we're only looking at binding regions on chromosome 1 only, so it doesn't really make an awful lot of sense, this particular output. There's lots of other pieces of information, for example whether the number of binding regions in the different promoter areas that we defined are greater than expected. By expected I mean over the reference genome as a whole. So we got we look at promoter regions, bidirectional promoters, which is where two genes on opposite strands share the same promoter, and looking downstream of genes as well. So you can just get an overall flavour of whether your binding regions are situated differently to what is expected in the genome. Of particular interest, and one that I'm usually quite interested in looking at, is where the binding regions sit relative to areas of a gene. And so again, this is the promoter divided into the areas we gave, different downstream areas which mirror our promoter setting, upstream, downstream, coding exons, introns, and distal intergenic regions, i.e. outside the 10,000 bases we defined as the promoter. And again, this compares it to what would be expected on the genome as a whole. So this could be quite interesting because it shows you whether you've got any sort of whether the binding events tend to happen in promoters, whether they tend to happen as distal enhancers. So it just gives you a bit of extra information about your genes, about your binding regions. And this shows you where the binding events are situated over the, well, in this case chromosome 1. Normally you'd get an output for all the chromosomes. It gives you an indication of the strength of binding in terms of the number of non-redundant fragments in those binding region areas. And then lastly, it gives you, because we supplied the WIG file, it's able to look and see whether these fragments are situated over different areas of the gene. So, for example, binding over the TSS, which is the transcription start site, the TTS, which is the transcription termination site, so the 3' end of a gene, and this analysis that allows it to look over 3,000 base pairs upstream, downstream, and over sort of normalized gene length. And then there's these plots to do with whether there's particularly more fragments over different portions of introns or exons. What you're better off doing is finding the description of CEAS, which is much more concise and well put than me talking in this video. Again, if you go to the shared data area, published pages, you'll see the basic ChIP-seq page that I've put there. Okay, so that's CEAS. When you run CEAS via the command line, you also get an Excel spreadsheet as well of all the RefSeq genes and where binding regions sit in relation to them. Unfortunately, at the moment this isn't available on our Galaxy instance, but it should be in the near future. This would then allow you to sort genes that contain binding regions, in particular gene regions that you're interested in, for example promoter regions, downstream regions, etc. Okay, so I'm going to end this video now on CEAS. Next time I'm going to be talking about how you can determine what sequences the transcription factor is binding to by motif discovery and a tool called Weeder. Until then, thanks, bye.